Okay, welcome to part 2 of the video lecture for chapter 6 on cell structure. You may recall that this was the slide I ended on in part 1 of this video lecture. We had just talked about the basic structures inside of a plant cell compared to animal cells. Now we're going to dive into what all this stuff means. What are these various organelles? How are they special? How are they related? What do they do? That kind of thing. We're going to start with the most obvious place, which would be the nucleus of the cell. Now, the nucleus, as far as a eukaryotic cell goes, is sort of like the command and control center of the cell, right? You don't find them in prokaryotes. You do find them in eukaryotes. And again, it houses the DNA. And because the DNA contains the genetic instructions that effectively tell the cell how to operate, that's why I say you can think of the nucleus as sort of like the command and control center, or the headquarters of the cell. It does contain most of the DNA in a eukaryotic cell, not all. As we'll see a little bit later on, both mitochondria and chloroplasts also have their own DNA. And we'll talk about why we think that that is a bit later on. So what we have here is a process of information flow in the cell. We talked about that a little bit at the end of the last chapter, chapter 5, regarding DNA and RNA. I said we had something called gene expression. Gene expression is the process that a cell uses to convert information as DNA into information as RNA and then ultimately into protein or amino acid sequence of protein. Gene expression is how the information in our DNA is used to build proteins. And so the nucleus containing the DNA is obviously a very important part of this process, as are the ribosomes, which we talked about as protein factories in the last lecture here, last video lecture. So the nucleus then contains most of the cell's genes and is usually the most conspicuous organelle. It's one of the obvious things you can typically see in a cell under a light microscope. It is surrounded by a nuclear envelope, which is the membrane of the nucleus, that separates it from everything else. And this nuclear envelope is actually a double phospholipid bilayer. So you see the nuclear envelope right there. It's actually a double layer, not really shown very well in this figure, but that just tells you something about how important it is to protect that DNA and keep it safe inside that nucleus. Also notice that the nuclear envelope is connected to the rough endoplasmic reticulum, which we talked about as sort of a protein factory, right? Because it contains ribosomes. Makes perfect sense that the rough ER be right there connected to the membrane of the nucleus rather than, let's say, on the opposite end of the cell, since the ribosomes are a critical part of making protein, as is the DNA. They're all part of this process of gene expression, so having them close together makes this process more efficient. And cells are nothing if not efficient. Okay, so also notice that we have these little pores that you see here in the membrane of the nucleus. Those are openings that RNA leaves through. Remember, the process of gene expression is taking DNA, or a gene, which is a segment of DNA that codes for a protein, converting it to messenger RNA first, as sort of a copy of the DNA. That messenger RNA then leaves through a nuclear pore, comes out, binds to a ribosome, and the ribosome converts its information to protein in the rough ER. So we have to have openings in that membrane for nuclear membrane for messenger RNA to leave. Now, if you zoom in onto an electron microscope view of the nucleus here, you can see some pores and that sort of thing, but zoom in and you see just how densely packed those pores are. These are pore complexes that allow messenger RNA to leave that nucleus containing the information of the DNA. This gives you some idea of how much information must be leaving that nucleus at any given moment. All right, finally, we talked about chromatin a little bit in the last video lecture, the first one for this chapter. Chromatin is another term for DNA, but specifically it's a complex of DNA, which you see here as this blue strand, and proteins. All right. Now, these proteins, which, by the way, the name isn't written here, these proteins are called histones. Histone proteins act like spools. They allow the DNA to wind around them, and that allows the DNA to wind up, coil up very tightly, very compactly, so that it actually fits inside that nucleus. There's about 2 meters of DNA inside the typical eukaryotic cell. That's about 7 feet of DNA. That's a lot, and it has to fit inside that microscopic nucleus, and the chromatin is a way for it to compact tightly enough to be able to fit. And so chromatin is a complex of DNA plus the histone proteins, right? Okay, so let's talk about ribosomes then a little bit more detail here. So ribosomes are not, first of all, they're not organelles. They do not have a membrane around them. They're not organelles. They're just really big molecules. Ribosomes are molecules that consist of, they consist of RNA, okay, I can write, RNA, in particular rRNA, ribosomal RNA, plus protein. So they're a complex of ribosomal RNA and protein, and they're typically kind of drawn sort of like this. They have two sort of subunits, a large subunit on top, small subunit on the bottom, but basically this is just one really, really big molecule, all right? You find them in two places in the cell. You find them in the cytoplasm, floating around. Those are called free ribosomes, but you also find them, as we've already said, on the rough ER. Those are referred to as bound ribosomes, okay? Free ribosomes and bound ribosomes. Okay, let's take a look. Let's look at the ones that you see in particular on the rough ER. So again, we're seeing the rough ER now highlighted in blue. If you take an electron microscope view of that, you can see these sort of jelly roll layers, right? These are the layers of the membrane. And look at all these dots. These are ribosomes. Now, how they know which ones are free and which ones are bound, you can't really tell from this image, but the point is that's a lot of ribosomes sort of embedded in the membrane of that rough ER. And again, this is how ribosomes are typically drawn, large subunit, small subunit. This is just in a computer model of what it might look like if you could actually see it. Again, no membrane around it, so it's not an organelle, it's just a big molecule, okay? But the sole function really of ribosomes is to produce protein. Okay, so we're going to kind of get off the topic of ribosomes here for a minute and shift over and talk about a large set of structures that sort of work together inside the cell. This is a collection of organelles basically that work together, but collectively we refer to these as the endomembrane system. Endo means inside, so this is internal membrane system, okay? And it consists of various structures. So first of all, we consider the nuclear membrane to be part of it. We certainly consider the endoplasmic reticulum to be part of it. Also, the Golgi complex, or Golgi apparatus, if you want to call it that. The lysosomes, which, as I said, are kind of like the garbage crews of the cell. Structures called vacuoles. We saw a central vacuole in a plant cell before. And then, depending on what author or authority you listen to, some people include the plasma membrane of the cell itself as part of this endomembrane system. I really don't, because endo means internal and the plasma membrane is the boundary between the internal environment and external environment. I don't really consider it an internal structure, but the plasma membrane does work in conjunction with these various other parts of the endomembrane system. Collectively, all of these structures account for a huge amount of surface area, of membrane surface area. This is a lot of membrane, okay? And in fact, in many cases, there's more membrane accounted for by the nuclear membrane, ER, Golgi, lysosomes, and vacuoles than there is in the plasma membrane itself, the outer membrane of the cell, okay? So these structures that we see here that comprise the endomembrane system, their components are either continuous, they're connected to each other. We saw the ER, the nuclear membrane, being continuous in the previous slide, or they can communicate back and forth via what are called vesicles. Vesicles are just little bubbles of membrane that bud off of one organelle carrying proteins or various other compounds, and they'll move through the cytoplasm down to the next organelle and fuse with it and inject whatever they're carrying into that next organelle. They're basically transport carriers, right? They're vesicles that carry things from one organelle to another, and that's a way for those organelles to communicate. All right, let's talk about the endoplasmic reticulum first. So we've already said that we have two types of this, the rough ER, which we've seen. It's called rough because of the ribosomes embedded in its membrane, and the smooth, called smooth because it lacks membranes. They're both looped and folded in different ways, but both ways are just mechanisms for increasing surface area. And again, that tells you something about the importance of these organelles. Lots of membrane means very important typically. We look at the rough ER first. Okay, so here they've shown you or they've cut away the nuclear membrane mostly. Nucleus was here, right? You see part of the membrane there. Here you see the rough ER in some cases attached to it, so it's continuous with it. And what you see here is a transport vesicle about to bud off from the outer surface of that rough ER. Here's one that's already budded off, and this is probably carrying proteins that will then go down to the Golgi apparatus, which would be down here, for example. Okay, remember that's like the FedEx of the cell, and so that vesicle will fuse with the Golgi and inject those proteins into the Golgi. We'll see more about that in a little bit. Over here, again, we have smooth ER. And the smooth ER, again, is sort of a system of interconnected tubules. That's another way to increase surface area. Smooth ER does not have ribosomes, so it does not make proteins. We'll see more about what it does here in a moment. Now, a couple of important terms. The ER lumen, whenever you see that term lumen, whether it's referring to this or some other structure in biology, it's referring to a void or a space inside, okay? The lumen of our digestive tract is the space inside the large and small intestine. Lumen means space or void or volume. This is the ER lumen. Okay, proteins fold inside that and then get packaged into vesicles. Cisternae. This term is just referring to these loops and folds of the membrane, these sort of flattened sacs that looping and folding produces. Those are called cisternae, and again, that's just a mechanism for increasing surface area. All right. Over here on the right, you see an electron microscope view of this. So let's imagine you took a scalpel and you sliced through this structure here and then looked straight down on it with an electron microscope. You would see this. Here you can see the cross-section of the flattened sacs, the cisternae, and you can see the ribosomes embedded in it, so that's rough ER. Right there's the ribosomes right there, for example. Here you see the sort of tubule-like structure of the smooth ER, what you're seeing here, right? So lots of surface area, made one way through the rough ER, made a different way or a different way of folding through the smooth ER. So what does that smooth ER do? Well, a number of different things. We've talked about what the rough does. It just does one thing, it makes protein. Smooth ER, first of all, synthesizes lipids. So it makes fats and oils. It's a membrane factory. It helps to produce phospholipids, for example, that are used to build membranes. Okay. Second thing it does is it detoxifies poisons or toxins. So in this way, you can think of the smooth ER as sort of like the liver of the cell, whereas your liver detoxifies potentially harmful compounds that you ingest in your food before it gets distributed to your body. The smooth ER essentially does the same thing for the cell, breaking down potentially toxic and harmful compounds that find their way into the cell. Finally, it stores calcium ions. Calcium ions are important, as are other metal ions for various metabolic reactions, and so the smooth ER stores up or acts as a reservoir for calcium ions that might be needed in various chemical reactions. Okay. All right, we've talked about the rough ER. It makes protein. We'll see more about how the rough ER works a little bit later on, but this is essentially the process here. So Proteins are produced in those ribosomes. They get injected into the lumen of the cisternae. They collect on one side of the membrane. It buds off as a vesicle, transport vesicle, which then takes them down to the Golgi apparatus, for instance. Okay, we'll see more about that here next, because next we're going to talk about the Golgi apparatus. So think about the Golgi apparatus as a series of flattened sacs. It looks very similar to the rough ER in the way that it's folded. The membrane is folded, and it does a couple of things. First of all, it takes in products from the ER, so it's taking in protein products. It's going to modify those products in a variety of fairly complex ways that we don't need to be concerned about. But most importantly, it's going to sort and repackage those proteins into transport vesicles that will then come off the other side of the Golgi. Now, I mentioned before that we have really two different types of ribosomes. Those we call bound, those embedded in the rough ER membrane, and free, those that are floating around in the cytoplasm, not bound to a membrane. Why do we have two different kind of categories of ribosomes? It's because the bound ribosomes are going to produce proteins that will eventually, for the most part, end up being transported to the outside of the cell. The free ribosomes produce proteins that will stay inside the cell and be used internally. So, when the bound proteins in the rough ER when the bound ribosomes in the rough ER make proteins and they transport them down to the Golgi is going to sort and repackage them and then ship them off to the cell membrane. Vesicles fuse with it and sort of boot their proteins to the outside, kick them out of the cell, where they end up in the bloodstream, for example. All right, so let's take a look at how that might happen. Okay, so here now we see the Golgi apparatus, right, just outside the rough ER. And here we have a transport vesicle containing proteins from the ER. It's going to fuse with this side of the Golgi. And notice again that we have flattened sacs called cisternae. So just like we saw in the rough ER, right? Okay. All right. Sorry guys, my phone is ringing. I'm going to ignore that. So we have these transport vesicles coming in. They're going to fuse with the cisternae, and they inject their proteins in to the Golgi in that way. Proteins will sort of migrate toward the ends of the cisternae. They're modified in certain ways, but when they come to the end of a sac or a cisternae, they can bud off into a new vesicle, which then may float down to the cell membrane. If we imagine the cell membrane is down here, that vesicle can come down and fuse and dump those proteins off to the outside. But this side where they come in from the ER is called the cis face, or the receiving side, okay? The side of the Golgi where they ship off from is called the trans face, the shipping side. That's essentially how this works in terms of getting those proteins to the outside. And so here you can see, in this electron microscope view, the flattened sac cisternae of the Golgi. You see a number of transport vesicles in the process of fusing, just like this one is, right here, to the cis face. And then you have a number coming off the ends and the transport side, the trans face, or the shipping side rather, the trans face of the Golgi, probably moving down. Maybe the membrane is down here on the south end somewhere. But that's essentially what's going on with the way that the Golgi works. Now, an organelle that's related to this process is the lysosome. Okay, so I mentioned lysosomes before. I said they're like the garbage crews of the cell. I said lysosomes contain hydrolytic enzymes. In other words, enzymes that run hydrolysis reactions. And so let's imagine that this transport vesicle coming off the transface of the Golgi contains hydrolytic enzymes. It will become a lysosome. Instead of fusing with the cell membrane and dumping its proteins off to the outside, it'll roam around the inside of the cell now and digest things that need to be digested. So lysosomes are produced by the Golgi complex. Okay, very important for maintaining, constant internal environment of the cell, breaking down particles that might get in, recycling worn out organelles, that kind of thing. Okay, in terms of a lysosome being used to break down particles that get into the cell, there are a number of different cell types that actually feed by taking in particles, okay? Think about maybe in high school biology lab you saw an amoeba or something like that, a single-celled protist that engulfs bacteria, other protists, or particles of food and digests them. Taking in a particle of food and digesting it inside the cell is a process called phagocytosis, also referred to as cellular eating. This is when a cell takes in a particle of food, and that particle will come in through the cell membrane. It'll be enveloped in a little bubble of membrane into a transport vesicle. Then a lysosome will fuse with that and digest that particle of food. That's what phagocytosis is. So lysosomes are very important, a very important part of the way that some cells actually eat. And here you can see this. So here we have a particle of food, bumps into the membrane folds inward, that's a process called invagination, pulls that particle in and wraps it in a food vacuole, a type of transport vesicle. Now you see that a lysosome comes along, fuses with that, and its hydrolytic enzymes digest that particle of food, okay? So it's one very important role that lysosomes play in the cell. Now, over here we have a process called autophagy. So again, phagophagy, that refers to feeding. Autophagy, self-feeding. So here we have a case where a mitochondrion or peroxisome, some organelles have worn out. They're no longer working, they're malfunctioning, whatever. The cell will envelop them in membrane to kind of separate them from everything else. A lysosome will fuse and digest those and recycle all those components. All right, so here I have a little video of this.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and moved through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Okay, so there you see basically how lysosomes are formed and the way that they work, all right? Okay, next thing to talk about are vacuoles. And I mentioned vacuoles before. They're again still part of this endomembrane system, but I also said think of vacuoles as a plant organelle. In particular, the central vacuole. A central vacuole is a big storage tank, a water tank inside that cell. In some cases, it can be upwards of 70 or 80% of the volume of the internal environment of the plant cell. In this case, you see it's so large that the nucleus, which is normally spherical, is sort of squished up between it and several chloroplasts here, okay? This is just showing you where it's located, generally in the center of the cell. You can't see it with the light microscope, okay, unless you stain it in very specific ways. And it's very difficult to see, but you can see evidence that's there because sometimes things are sort of squished up on the inside of the cell membrane as a result of that large vacuole taking up so much space. So this is one of the major kinds of vacuoles that you see in plant cells, and there are a variety of other vacuoles that perform various different functions in different kinds of cells, but generally they're for storage or something like that. We also have food vacuoles, okay, for one. So we've already seen some examples of that with regard to phagocytosis. So food vacuoles are formed by phagocytosis. We have another type called contractile vacuoles. These sort of act like bilge pumps in a boat. A lot of single-celled protists that live in freshwater have these. A freshwater organism has one sort of overriding problem. Water is constantly flooding into its internal environment. And if it's a single-celled organism, that could cause that cell to rupture, like a balloon you put too much water in. So the contractile vacuole will sort of absorb that excess water, and once it gets full, it bails it to the outside. It gets rid of it, like a bilge pump would in a boat, for example. And then, like I said, we have central vacuoles, like I showed you in the case of plants, okay? These are really the major categories of vacuoles. All right. So here we have basically a review then of the endomembrane system. So it consists of the nuclear membrane, which is continuous with the rough ER. We have the smooth ER over here. We have the transport vesicles carrying ER products down to the Golgi. We have transport vesicles carrying products from the Golgi down to the cell membrane. We have lysosomes. All of that is the endomembrane system. It all kind of works together to manufacture products, ship them off to the outside, so on and so forth. All right, I think the last two that we'll talk about today for the second part of this video lecture are the mitochondria and chloroplasts. So the mitochondria and chloroplast are the energy-related organelles, okay? What mitochondria do, let me just write this out here. Mitochondria carry out a reaction called cellular respiration. So basically what happens here is that when you ingest food, whether they're proteins, sugars, fats, or whatever, those food molecules are transported to your cells through your digestive and circulatory system, and your cells take them in. Your cells break them down to extract the energy from them. That's what cell respiration is all about. So and for the equation here, we generally show it as a glucose molecule, sugar, but you could also have a protein or a fat, as we'll see later on. But essentially we have C6H12O6, okay, so 6 copies of that, plus 6 oxygens, and we produce 6 CO2 plus 6 water plus energy. E with an up arrow means energy. So this is cell respiration. Don't worry about it too much right now. It just means that sugar is combined with oxygen and it's broken down to CO2 and water, and energy is given off, and the cell captures and uses some amount of that energy, usually about 40%. It's captured and used. That's what goes on in the mitochondria. Now, in the chloroplast, we have photosynthesis. So again, this is the process that plants use to turn CO2 and energy from sunlight and water into sugar molecules. So what's going on there? Well, a plant in its chloroplast will take 6 molecules of CO2, combine it with 6 molecules of water. In the presence of light energy, it produces 1 molecule of glucose and oxygen as a byproduct. Okay. So here we have a process that basically is going to convert CO2 and water into a food molecule and produce oxygen as a byproduct, okay? And by the way, that 6 there is not right. Sorry about that. And we do have a 6 there. All right, so if you look at these two reactions, you'll see they're the reverse of each other, where mitochondria takes sugar, combines with water, converts it to CO2 and water. Sugar combines with oxygen and makes CO2 and water and gives off energy. Chloroplast combines CO2 with water in the presence of energy to rebuild sugar and produce oxygen. So that's what these two organelles do. This is why they're called the energy-related organelles, okay? And we'll see a lot more of the details of these reactions a little bit later on. Now, let's talk about mitochondria first. So mitochondria basically look like kidney bean-shaped organelles. They have an outer membrane that's smooth, and they have an inner membrane that is highly looped and folded. It's in that inner membrane, you see right there, where a molecule called ATP is produced. So this energy up here, about 40% of it is packaged into ATP molecules, and that ATP is produced in that membrane. That's why you see that membrane looped and folded. The more that membrane looped and folded, the more copies of the cellular machinery needed to make ATP, and so the more ATP that can be made. Our cells, on average, need to make about 10 million molecules of ATP per cell per second. That's a lot, and so we have to have a lot of membrane to be able to do that. Okay, So let's look at the structure of a typical mitochondrion. Okay, so here's essentially what I drew in the previous slide. So we have our outer membrane smooth, right? Inner membrane looped and folded. And now these loops and folds we see are called cristae. Okay, it's just a way to increase surface area. That's where the ATP is produced. We have a fluid-filled space inside of that called a matrix, right? This is the fluid-filled space. Inside that we have ribosomes and circular DNA molecules. Okay, what does that look like to you? Think about it. It looks like a bacterium. And there's actually very good evidence that mitochondria, at an early point in the evolution of life on the planet, used to be free-living mitochondria. The idea is that you now have them in the eukaryotic cell, so how'd that happen if they were free-living bacteria at one point? Well, at one point you had a proto-eukaryote, let's say maybe 2.7 billion years ago, something like that, and it consumed a bacterial cell through phagocytosis. And for whatever reason, that bacterial cell, once it went into the larger cell, was not digested, and it became a part of that cell. So the cell that ate it now has something inside of it, a new structure that can make large amounts of ATP. That's a distinctive evolutionary advantage. What evidence we have for this? If you look at the structure, it looks like a bacterium, but if you look at that DNA is very similar to bacterial DNA. The DNA sequence, okay, there are bacterial genes found in that. So it's very strong evidence that these guys really did used to be bacteria. That process I talked about, whereby this early bacterium was consumed by a proto-eukaryote, that is called the endosymbiotic theory. Endosymbiotic theory, okay? So the idea is that these bacteria, which are now mitochondria, formed a symbiotic relationship, a mutual relationship with the cells that consumed them and became part of them, a functional part of them. All right, take a look at this electron microscope view. You can really see the cristae very clearly here, right? All these little dots you see between are the ribosomes. Those ribosomes are used to make proteins from the DNA inside of this. They're self-contained units, again suggesting they were bacteria at one point. But you see how many ribosomes are there, okay? All right, what about chloroplasts? Well, they're next. Chloroplasts contain the green pigment chlorophyll. Okay, let's go ahead and look at the figure there. So this is a chloroplast, and again, they contain the green photosynthetic pigment, chlorophyll, and basically they're put together in a very similar way to mitochondria, multiple membranes, in other words. But here we actually have an outer and an inner membrane that are both smooth. Inside that, we have a third set of membranes, again, highly looped and folded, but they're looped and folded in such a way it almost looks like they're like a stack of dinner plates, okay? Each stack here is called a granum, right? So that's a granum, for example. The individual sort of, quote, plates of a granum are thylakoids. So that's a thylakoid. There's one. If you look inside, they're actually just looped membrane, but from the outside they look like separate structures called thylakoids. This is the thylakoid membrane. That's where the chlorophyll is found. And again, the chlorophyll is what absorbs sunlight and helps to convert it to energy that the cell can use. Okay, we also have a fluid-filled space around that. The fluid-filled space is called a stroma. You can almost think of it as cytoplasm. It essentially is. And you have ribosomes. You also have circular DNA molecules. So again, we see chloroplasts, like mitochondria, have their own DNA, and that DNA consists of bacterial genes. Okay, very similar to modern bacteria. So again, we think the same thing happened here, endosymbiosis. We think at some point an early proto-eukaryote engulfed or phagocytized a photosynthetic bacterium, didn't digest it, and that bacterium became incorporated into that cell structure and began to produce sugar for it. That cell now has something inside of it that can produce sugar in the presence of sunlight and CO2. Very adaptive, very beneficial. And again, how do we know? Why do we say that? These guys look like bacteria. They have their own DNA, their own ribosomes for making proteins from their own DNA. Very, very similar to bacteria, modern bacteria. And the genes that they have really scream bacteria. The gene sequences are bacterial. This is an electron microscope view over here. You can see the outer membrane, maybe not the inner membrane that's smooth, also just inside of it, maybe right there a little bit. But here you see a granum. There's a second one, third one, and the thylakoids make up those grana, but lots of surface area there. And the stroma, again, is the fluid that all this is sitting in. Okay. Chloroplasts are a member of a category of plant organelles called the plastids. All right. Plastids are plant organelles that have very specific functions. A chloroplast is one type of plastid. There are others that we'll see a little bit later on. But plastids have very, very specific functions. Chloroplasts really do one thing. They run photosynthesis. They make sugar for the plant as long as CO2, sunlight, and water is present. Okay, so those are the energy-related organelles. And we've seen how they're related, right? They're related in the sense that mitochondria break sugar down and produce CO2 and water. Chloroplasts take that CO2 and water in the presence of sunlight and rebuild it back into sugar. So it's this sort of cyclical chemical relationship between these two very important organelles. Okay, we're going to stop there. We'll pick up with the rest of this. Hopefully we'll finish chapter 6 in the third video lecture coming up.